Physical reason for it. And your mic's on.
Yep, it is. Okay. So I'll give you an introduction to what Burkhard already briefly introduced, how to predict the 3D structure of proteins just from sequence using evolutionary sequence variation, going from 1D through the intermediate step of 2D prediction to 3D prediction of protein structures. And so this lecture has 3 parts. The first one is a short prologue about the basic idea that underlies this work, how you predict 2D contact maps of proteins using correlated residue exchanges. And then in the second part, I will go into a bit of the technical details and the math of how you do that. So what are the algorithmic approaches to extract these residue contacts. And in the third part, that will be the last step, going from these predicted contact maps to full 3D structures of proteins. And so if you have questions, just interrupt at any time. If you don't understand something, or if you feel I should go into detail a bit more, just let me know. So the very basic problem is to go from genotype to phenotype, which is basically all of biology, so it's terribly general. In this case, it's going from the DNA sequence of the protein that directly gives the amino acid sequence of the protein, going from that amino acid sequence to the 3-dimensional structure of the protein. And why is that an important question, or why is it an important problem that people want to solve? It's because these 3D structures, as I suppose you've already heard in the lecture, are pretty hard to determine experimentally. So these are very costly experiments that take a lot of time, often just don't work because of some often unknown issue. So it's very helpful if you can at least get a rough idea of what a protein looks like using computational methods. And so this man's work lies at the basis of what I'm going to talk about today. This is Christian Anfinsen, and he won a Nobel Prize in 1972 for showing that all the information that you need about a protein structure is encoded just in the amino acid sequence. So you need only the amino acid sequence, and that alone contains all the necessary information. There are some caveats, but in general this still holds true. And there's, coming back to what I just said, that structures are very hard to get experimentally. There's a thing called the sequence structure gap. The number of sequences that we know from genome sequencing, from thousands of different bacteria and eukaryotes by now, we know lots and lots of sequences, millions of them. By now, I think the number in the UniProt database is about 80 million sequences. And it's just the... there was a drop again, right?
Exactly.
It's very odd.
There's a simple reason. So the simple reason, sorry that we take this discussion, but the simple reason is, so they had 85 million sequences in the database, and then they decided that everything that is 100%, every pair that is 100% sequence identical will be merged. And that got us 55 million. So that's 30 million somehow duplicate records.
Anyway, it's 50 million still allowed. And so the number of...
So you said identical, which means purely identical, the sequence? Or?
Yes, but now so the word identical has, I'm sorry. So the word identical, thank you that you ask, because the point is, identical could be identical over the entire length. So the entire sequence is 100% identical. And that's not the case. It is essentially back to the problem of protein being parts of other proteins or being made up of units. So yes, identical, really 100%, but fragments sometimes. And that's why they were not recognized. So you have something that differs somehow from what you had before, so you open a new record. And there's a logic for that. But then there's also a logic to say, well, if it is 100% identical, although it's shorter, then for that stretch is somehow a segment that should belong to the other one. And actually, in some cases, maybe they exist as two different entities. So,
yep.
Sorry for getting into that detail. But
Yeah.
made a mistake.
But so no matter if it's 80 million or 50 million, it's still orders of magnitude between the number of structures, which are right now I think about 100,000, which are highly redundant, so they're often the same protein over and over again anyways. There's a high gap between the number of sequences that you have, or if you group them together, if they're similar in terms of sequence families. There's a high gap between what you have in terms of structures and what you have in terms of sequences. And you want to know for lots of these sequences, what do the proteins look like? But the gap between the number of sequences and the number of structures just hasn't closed, even though these structure experiments have gotten faster and more efficient. So clearly, to close this gap, you need computational predictions. And so this work that I'm going to present is kind of turning it around a bit. It makes actually use of all these sequences to get information about the structure. And this is how it works. It's a very simple idea in principle. So let's just assume this is a protein, and these two blue spheres are residues within that protein. Of course, you have lots more. And in the three-dimensional structure, these two residues are in contact with each other. They touch. And of course, you've got many more of these pairs that spatially interact with each other. So they kind of glue together and define what the three-dimensional shape of the protein is. And it's basically the chemistry of these amino acids that interact with each other that stabilizes the folding of the protein. So for instance, you could have this one residue is positively charged and this one is negatively charged, and it's this positive-negative charge interaction that is favorable. Or you can have two very sticky hydrophobic residues, and the hydrophobic interaction between those stabilizes the protein. In any case, it's kind of an interaction. This is just a schematic depiction. It's an interaction between two residues that somehow fits together, that is kind of optimized for each other. But now what happens, or what can happen, is that there's a mutation in the protein sequence. So one of these amino acid residues changes to a different residue type. So let's say this one was positively charged, this one negatively charged. Now all of a sudden you've got an amino acid that's not charged at all and a negatively charged one. So this is not a favorable interaction chemically anymore, which destabilizes the protein. So it's maybe not as stable anymore, or it doesn't fold into the proper three-dimensional shape anymore. But so to recover, what can happen is if there's a second mutation in the other residue that restores that favorable interaction. So if this negatively charged amino acid now goes to a hydrophobic amino acid, you get not a positive-negative charge interaction anymore, but just two sticky bits sticking together, for instance. And once this compensatory change, it's called a compensatory or correlated mutation, occurs, you got a functional protein again. Or at least you got a more stable protein again. And the whole idea of this approach is you try to look for these exchanges that occur together. So you have a protein structure, and if you, for that protein, create a multiple sequence alignment, so you collect all the related sequences in the sequence database, and you look at those two positions in the protein, for now, assuming you know the 3D structure and you know that these two residues are in contact, what you observe in the multiple sequence alignment, so each line is one sequence, what you observe is coordinated, correlated exchanges between the residues in those two columns. And that's pretty easy because you already know the answer. So this is just the concept. What's more interesting is, can we go the reverse direction? Can we start from sequences, which I just showed we have lots and lots and lots. We're just drowning in the sequences. Can we just take the sequence alignments And in that sequence alignment, look for correlated residue exchanges, and therefore solve the inverse problem. Can we figure out from these residue exchanges which residues are in contact in the three-dimensional structure? Because once you know which residues are in three-dimensional contact, you can use that information, it's basically the triangle inequality, and say this residue is close to this residue is close to this residue, and this defines the three-dimensional shape of the protein just from the information which pairs are close. And so going from here to here is actually pretty straightforward. Once you know which pairs are close, going to the three-dimensional shape of the object has been well established for 20 or 30 years from structure determination in NMR. Yep.
This is by knowing only these two out of many, right? In the sequence. You have information not for the whole sequence how they are matching, but just for a couple of them which were correlated.
Yeah,
right?
a subset. So it's of course more than two.
Yeah.
So let's say if we have a normal protein, we would talk about 100 or 200 of those pairs. And they are scattered throughout the entire 3D of the protein. Yeah?
So I didn't get it good, I think, because you said that we're using the chemistries of the residues that are in contact. But you said even that when a mutation is occurring, we can lose this chemistry, like with the negative and the non-charged one.
Yeah.
So another mutation should occur, and we should have like no charge, but since they are lipidic, they're sticking together. But can we recognize even this thing? Like.
That's the challenge.
That's the challenge. That is the very challenge. So it's just looking, and I will have a concrete example of that
So
a bit
I
later.
have to say something else here that I didn't say clearly before. I'm sorry, I should have introduced contact prediction. And if I had introduced it, I would have given you 20 years of failure. Thomas is going to show you 3 years of succeeding.
Okay. So I'll go into the details of how it actually works in a bit. But the basic principle is clear to everyone? Okay, great. So, yeah, there are the 3 question marks. How do you actually do it? And that leads to the second part, which is what are methods to figure out which pairs correlate, or the word we are using for that is co-evolve, because they change together over the course of evolution. And the first idea is very simple. That's basically the one that I'm going to spill the answer already. These are the approaches that pretty much failed, but they got the basic idea right back then. So this is a sequence alignment. Each line is a sequence, let's say of the same protein. Let's just assume this is whatever, hemoglobin in different species like human, cats, cow, whatever. All the protein species that have a hemoglobin. And so let's look at the red columns here and here for now. And so you have a negatively charged amino acid here, aspartic acid or glutamic acid. And over here you have a negatively charged amino acid, but there are different ones, arginine or lysine. Oh, positively charged, thanks. And a very simple idea to see if there's any correlation between the 2 columns is what are the frequencies of certain amino acid types in that column. In the one column, column I, in the other column, column J. And then if you look at the frequencies of the amino acids in the pair, so Fij, how do those pair frequencies relate to the single column frequencies? So for instance, the frequency of D would be 0.5, the frequency of E would be 0.5, here the frequency of R would be 0.5, and of K would be 0.5. Let's see. But so the frequency of D and R together is 0.5, and the frequency of E and K together is also 0.5. And now the question is that we ask is, to what extent do the pairs deviate from what you would expect under statistical independence? So what's the difference between the pair frequency and the product of the single frequencies? Now the single frequencies for each pair would be 0.5 times 0.5. So what you would expect if these columns are independent, that the pair frequency is 0.5 for each of these two pairs. But in both cases, the actual frequency is 0.5. So there's a difference of 0.25 between what's expected and what you actually see in the sequence alignment. And that suggests, because this is different from 0, these columns are not statistically independent, and therefore these two columns in the alignment co-evolve, and we would assume, because they co-evolve, they are close in the three-dimensional structure. And if you want to quantify that a bit more in a theoretical sense, in terms of information theory, the quantity that you would calculate is called mutual information, that measures differences between distributions. So that's all over statistics. You look at what's the difference. Now it's not the difference, but the ratio. What's the difference between the ratio between the pair frequency and the product of the single column frequencies? You take the log of that, and you also weight that by the pair frequencies. That means you give more or a stronger weight to things that you see more often, and therefore you believe more strongly because you just see it more often, and it's maybe not just some spurious random association. And you see the sum here, which runs over AI and AJ. These are just the 20 amino acid types. So AI would go from alanine, cysteine, glutamic, aspartic acid, and so on, over all the 20 amino acids. And AJ would also run over all the 20 amino acids. So you have 400 numbers in total that you're summing up. So if we jump back. In this particular case, you would just see those two types of amino acids, and all the other possible pairs you would just not observe in the data, and therefore they wouldn't be in the sum. But in principle, you go over all 400 amino acids. So what this gives you in the end is a single number for any pair of positions in that protein, I and J. So you have a certain protein, and you can ask what's the mutual information between those two residues. And so you will get pairs that have a very high number. Those are the ones where you think these are correlated or they're co-evolving, and you will get some, at least that's what you would expect. If you have a low number, then there is no correlation between the two columns, and therefore you wouldn't assume that they are in three-dimensional contact, or at least co-evolving. Now the bad news, and that's what the last 20 years basically showed, is it doesn't work. I'll just reintroduce what contact maps are. Did you ever talk about those before today? So I'll just explain it again, because we're going to see those more often in this talk. So what you see is a contact map representation of a protein. It's a 2D representation. What you have along the axis is the protein sequence, and on the other axis it's also the protein sequence. So each point in this matrix corresponds to a pair of residues. And if you have a gray dot in here, that means those two residues in the protein structure are in three-dimensional contact. So you say they are closer than some distance, in this case 5 angstroms. And a blue dot now means this is a pair where we calculated from the sequence alignments that they're very correlated. So they have a high MI number for this particular pair. And it's the top 100 pairs in this case. And what you see, or what you would hope, is that the blue dots would fall on top of the gray dots. So the pairs where you say they correlate, they actually correspond to pairs that are close in the three-dimensional structure. Because if they're not, the whole idea falls apart, because you can't get the structure this way. Now,
it shouldn't follow the diagonal, right? the blue dots are not supposed to go on the diagonal. The diagonal is because we're the same element on the same element, right?
So the very diagonal is just same. So there is no contact as such because it's the same residue. But if you go just one step on either side of the diagonal or two steps, this is the chain of the protein, it's the backbone. So because it's along the chain, these residues are in contact almost by definition, at least the ones that are right next to each other. And because they're next to each other, you would actually also expect blue dots that run along here, because you would expect that neighbors in the sequence, because they are close, also correlate over the course, or co-evolve over the course of evolution. But yeah, so these points cluster very much at the two ends of the sequence, and there is a handful, like 3, 4, that are in this region that do fall on the gray. But what we would need is, because these are the pairs that really define the global overall 3D shape. The chain along here is pretty boring, you know that anyways, because that is the protein sequence. But it's these that really give you the information what the three-dimensional shape is. So what we would need is lot more points, a lot more blue on the gray in this region of the plot.
Let me just draw your attention to 2 points here. One is, mostly this map is pretty thin. So most of the contacts somehow are along the chain, but actually most of what you see there is white. So the further you go away, so somehow this implies that structure formation is determined by a few what people call long range, so distance in sequence contacts. That is not trivial. And you can argue why that is the case, but this is one observation here. The second thing, I'm not entirely sure whether you clearly said that, this is trivially completely symmetric with respect to the diagonal. You said that?
Yeah, I didn't. Yeah, and so it's not only that the points don't fall on the gray, actually they are, or that we lack coverage of the gray, most of them are where no contact is, so this would be totally useless information for getting the 3D structure. And to just come back to what you just asked before about the chemistry. So right now, this approach only uses the letters, so it just looks at the 20 letters, and it doesn't explicitly incorporate information about charge or size of the residue. There is work going on that also looks at the actual chemistry of those residues, but for now it's just the information about the chemistry that's encoded in the letters co-occurring or not. So it's very implicit in that sense. So what's the reason it doesn't work? The main reason it doesn't work is transitivity, and what that means you'll see here. So this is again a protein, but now I'm highlighting more than 2 residues. We have 4 residues, I, J, L, and K. And let's say this residue changes. And that destabilizes the protein, or it loses its function. Then this residue I will need to compensate for that first mutation. What that in turn will cause is that L changes. And then, because L changes, K changes. So this change in J triggers this whole chain of changes throughout the molecule. Because all of these pairs are very tightly together and tightly packed, you will observe these transitive effects, and you will see a change in J, and you will see a corresponding change in K, but they're not close. It's just that these changes propagated. And so what you have is, if you look at all the correlations, what you will observe is you observe the actual changes or the actual correlations, so kind of the cause. But on top of that, you will see all these transitive effects that are just induced by this chaining. And what often enough happens is that actually these transitive effects are stronger than the direct effects, because it's not just this regular chain. If it was just a regular chain, you could figure it out pretty easily. but you got all these pairs everywhere, and there's so many different combinations, things can change, that the indirect change might overpower the direct change, and that phenomenon is called superadditive correlations. So what we really need is some approach that can figure out what's going on globally, because if you only look at one pair at a time, you will never have a chance of figuring out if these are direct effects, or if this is the direct effect, because you know nothing about the context of the whole problem. So what needs to be done is you need to look at all these pairs at the very same time, and if you look at all the pairs at the same time, you might have a chance of figuring out that actually these are the direct effects, and what you observe between J and K is only some sort of indirect correlation. So what we're trying to do is we go from observed correlations and try to reduce these observed correlations that have all this noise and this false signal in there, to go to the direct causal coevolution between positions in the protein. And the way to do this is called inverse Potts inference. It's a term from statistical physics. So this basic problem has been known for decades in physics, but biologists just didn't think of applying the techniques that had been developed in statistical physics on this biological problem. There is a second name for this. It's what we're using is an undirected graphical model. So that's more in the statistics community. It's also been well known for years and years that you can do this, and it's just been sitting there waiting to be applied to this particular problem. Now, how does this work? The idea here is that you use probability model that connects observed data to some underlying model that consists of direct interactions. And so the goal is you go from data that you observe to figure out what the parameters of this model are. So it's kind of a twisted logic. Usually you have a model, and that model generates something for you. But this is a parameter learning process that we're doing here. So you take the observed data that has all the indirect effects in there, and you try to figure out statistically what is the model that generates, or that generated all these indirect effects. So you're going backwards in a statistical sense here. And why is it this particular model? I will not go into the details of that. You can show statistically. This is just the simplest model for this particular problem for the jobs that has the least assumptions about the problem. So it's called a maximum entropy model. You could come up with other models, but they would make additional assumptions, and because we don't really know what the best model is, we just use this one. So it's the simplest model. Like the Gauss distribution is the maximum entropy distribution for a set of data that has a variance and a mean. That's kind of the same idea. You could have more complicated distributions, but that's the simplest one. And the way to go from here to here is basic statistics. You perform approximate maximum likelihood inference. So you have some data, and from that data you try to learn the parameters of a statistical model. Is everyone familiar with that concept, or do I need to go into a bit more detail here?
Yeah, maybe.
Okay, so I assume then not everyone has heard a statistics lecture in here, right? Okay, so it's I'll keep it very general. It's on the next slide. So what you have some sort of data. In our case, that's the multiple sequence alignment, and you're trying to figure out what are the parameters for that statistical model that best explain the data that you have. So you're trying to find the most likely model that explains the data that you have, the sequence alignment. So what you do is you just write down the probability according to this probability model I just showed on the previous slide. And of all the sequences in the alignment, so it's just a product over the probability of each sequence, given some parameters. And now this whole thing is a function, because you already have the data, it's a function of the parameters. And so you write down this whole thing, you differentiate with regard to 0, and you try to find these parameters h and j that maximize this function, which are the parameters that best explain the data. And it's these parameters that we want. These are the direct pair interactions between positions in the alignment. So these are conceptually the numbers we're after, because they would explain the direct change between pairs of positions in the alignment without the transitivity, because going from here to here, the transitivity just gets cleaned out. And so you have one of these numbers, Jij, afterwards, for each particular pair of amino acids, let's say D and K, between a certain pair of positions, I and J. And because we don't care about the particular amino acid type in those positions, we just sum all these numbers. And we get a single number again, like the mutual information number that I showed before, that quantifies the strength of coevolution between the two positions. But now this number has this transitivity and some other problems cleaned out, and it's kind of the direct causal coevolution between the positions, not the raw correlations anymore. There's some other technical details I won't go into in calculating these things. If you're interested, there's 2 papers that you could read that really lay out all the math. That's Marx et al. That's the first version of the algorithm. Then there's also Eckeberg et al., which is the one I would recommend to read, that gives all the details of how you solve this problem in a very understandable way with some statistical background. But now the major question is, does this cleaning up and all the effort that we invest, does it work? And as it turns out, this is what we had before, the mutual information didn't work. But now if you look at the solution that we get out of this global model, so these Fnij numbers I just showed, you see that the red, in this case it's now red dots, are mostly on top of the gray dots, and they are nicely spread throughout the entire contact map. So the statistical inference process.
Can you turn around? Can you stand here?
Oh, yeah.
Because then you only stand in my way now.
Okay, sorry. Oh, no.
I can move to
Oh, it works like this. Yeah. So what you see here is these pairs correspond to the experimental 3D structure contacts. And now, in this case, I think it's pretty intuitive that there might be hope of getting the 3-dimensional structure back out, because you know what is close in the 3-dimensional structure. Yep.
Oh. The idea was good.
Okay. And this is a funny side note about the whole thing. So Burkhardt just said people tried this for the last 20, 25 years and didn't succeed. And it was only in the last 3 or 4 years that people started using these global statistical models to solve the problem. Well, actually, no. He did it before everyone else. This is, oops.
Sorry.
Oh, this is Alan Lapidus, who did his PhD with Stephen Hawking. And he already solved this problem in 2001. And the paper got never published. And it set back an entire scientific field for more or less 10 years just because the journal Scient didn't publish his results. And people didn't believe him back in the days. So in a way, all the credit for this belongs to him because he figured it out before everyone else. Now, we know we have pairs that are close in 3-dimensional structure. Can we get the actual 3-dimensional structure of proteins out of that? And this is what the third section is going to be about. And so the question is, what can we predict from these pairs? We can predict the three-dimensional structure of proteins, so single protein molecules. What you can also predict from these contacts between residues is protein complexes. So once you have more than one protein, and these proteins interact with each other, the same logic applies. So it's also the chemistry between different proteins that maintains interactions between proteins, and therefore you will observe coordinated residue exchanges. But first I will talk about single protein molecules, and just as a brief reminder, so we know how to go from here to here. And now the question is, can we successfully go from this pair information to the three-dimensional structure? And this is the first time it's been ever tried. This was in 2011. And so this was the very first attempt of using, successful attempt of using coevolution to predict three-dimensional structure. And here are 3 examples where it's worked pretty well. So these are 3 different proteins that are predicted using this approach of using coevolution. And these are the experimental structures. So these have been crystallized and solved by X-ray crystallography. And as you see, just visually, there's quite high agreement between what you see on the left side and on the right side. And you can also quantify this properly and show that these are very good predictions that you just get from the sequences using nothing else really of the three-dimensional structure of the protein. And that was really a huge breakthrough. And usually people think about this three-dimensional structure problem, maybe you've heard about it before. You need really big computers, and there's this thing called Folding at Home or Rosetta at Home, where people invest lots and lots of computing power to figure out these three-dimensional structures. And it kind of works for very small proteins, let's say of this size, but you would never ever get out the structure of a protein that's as big as this one. And so that was kind of a shocking revelation, because all these results were done on a MacBook Air in 2011. So it was really a paradigm change of how you approach the problem, and at the same time the first time it successfully worked. Okay. And then the next step was, and this is where my master's thesis comes in, was to figure out, can we also predict membrane proteins using the same method? The thing about membrane proteins is they're even harder to get the structures by experiment, because they are sitting in this lipid membrane, and once you take them out of there, they don't have their three-dimensional structure anymore. So there's much less information on experimental 3D structures of membrane proteins. And at the same time, they tend to be much bigger on average than regular proteins. So it's a twofold challenge, because they're bigger, which makes the problem harder mathematically. And also there is fewer information available. But for the cases where we know the answer, we could show that it is possible. And now you see the experimental structures in gray, and the predicted structures are overlaid in blue. You see that the fold that you get from the prediction also corresponds pretty well, except for details, with the experimental structure. So there was also quite a big breakthrough, because for the first time you could just get these structures from sequence without running expensive experiments or investing lots of compute time. Now I'll also go into the second part very briefly, which is to predict protein complexes, which kind of generalizes the whole approach by one step, because you don't look at single proteins anymore, but you look at pairs of proteins. So at the same time you look at what's going on within the protein, what are the closed pairs within the protein, and also what are the pairs that interact between the two proteins. And so that's what I just said. So you have a protein A, you have a protein B, and they touch in a certain part. And so you would expect that these residues in the one protein change in a coordinated way with the others. So if you change this residue and they don't fit together very well anymore, that might abolish the interaction between the proteins because they don't glue together in a simple sense. So the idea in this case is to figure out what are the residues that change together between the two different proteins. And it's pretty much the same idea. There are some really complicated technical details which I won't go into. But all you do is, instead of looking at a sequence alignment now of a single protein, which you would have here, you need a joint sequence alignment of the two proteins together. So you need a sequence alignment of protein A, you need a sequence alignment of protein B, and you have to bring those two sequence alignments together into one big sequence alignment and run the same statistical analysis. And what that gives you in the end is which of these pairs are interacting, and that you could use to figure out, knowing the two interacting proteins, what is the exact three-dimensional way of how they interact with each other. And so these are also experiments that are very hard to do, in a way even harder than figuring out single protein structures. So using this computational approach here can also help figuring out what protein interactions look like without investing lots of time into experiments that might not work out. Yeah?
So to see the interaction between two proteins, we're not using anymore the calculating the pair interaction. So we're using more columns at the same time.
Yeah. So you're just pretending it's, at that point, you're just pretending it's one big protein. You put the two interacting proteins together into one big protein, and because you know where the boundary is between the two, then you can say...
Yes, I understood this. My question was about using the pairs. Even before, we were using just pairs, now we're using more, or...
No, it's still just pairs. It looks at all the possible pairs at the same time. And it's a good question. So you could also use triples or quadruples or whatever. In principle, you could go as high as you want. The problem is, the more once you...
It's a figure. You show a triplet. Maybe that could be...
Oh, I see. I see. That is actually very confusing.
Yes, exactly.
Good that we published it this way. No, it's still just pairs. So yeah. That's what you figure out 2 years after making the figure.
in both.
Yeah.
You do it on both, right?
Yeah. So it's still just pairs that we're looking at. But as a side note, you could use triplets, for instance. But by doing that, you have many more parameters in your statistical model. And so we have many sequences, but we don't have that many sequences that we don't run into bad overfitting problems if we use triplets. And this is now one of the examples in detail. So this is a membrane protein here, and that transports stuff across the cell membrane. And this is a second protein that interacts with this transporter protein that kind of gives the energy to this protein to drive the transport. And what you see here again is a contact map representation. It's slightly more complicated because it shows both the contact map within the protein. So I just left away the second half because it's symmetric. And here again, that's the second protein. And what you have here in this quadrant, these are the interactions between the proteins. So any dot in here says one position in the first protein is interacting with one position in the second protein. And as you see again, the colored dots are pretty much on the gray. So the coevolution analysis gives you the pairs that do change between the two protein subunits.
And this dotted one is that dot over there, is it? There is one red line in the 3D structure.
Oh yeah.
Is dotted.
Yeah. So that's the one that's wrong. So the dotted one means it's distant in three-dimensional structure. Yeah. Well spotted. And so you can apply this to many of the complexes where you know the experimental structure, so where you do know how they interact. And you see that a lot of the pairs that are identified between the proteins do corresponds to actual three-dimensional contacts, and the ones that don't are again in dotted lines, but you can see that, so I'll just claim they're all right. And of course they're not, but overall you can reliably figure out how these proteins go together to form the protein complex just from sequences.
So I'm supposed to see some dotted lines?
Yeah, but you don't see them.
Okay. Fair enough. Great idea.
And again, what you can do is you can apply this now, because what I just showed you was boring because you know the answer. So that's just method development. But can you apply this method to figure out something about proteins where you don't know the interaction? And so these are now cases where we know what the single proteins look like, but we don't know how they interact. And so these are predictions. No one's ever seen the interaction between those before. And those lines suggest what the interfaces are, and they're biologically plausible because they cluster in certain parts of the molecule. And in some cases, they also agree with what's known about the protein interaction from other experiments where people have, for instance, cross-linked the two molecules and see in which parts they stick together, but not really knowing what the three-dimensional look of this is, just having kind of point information, what the interaction is, but not really knowing the full level of detail. Yep. And so if you want to try this out yourself on your favorite protein of interest, we have a web server, which is at evolut.org, and you can put in, you should just select this button, evolut, and you can put in your protein sequence of interest, and hopefully it gives you back a good answer. And so if you want to try it, you can just use a protein where the answer is known, and you can put in the known answer, and you get all the same type of analysis I just showed you with the gray dots underneath the colored dots. And yeah, so there's this other thing for the complexes, which is called EV complex, which you can't read here. It says under development, which will hopefully come in the next month, but that's still to be done. Yep. One more or two more. And if you want to read up anything about this whole story, these are the papers I talked about. So this has been the string since 2011, 2012, 2014, and it gives all the full stories on each of these topics.
There are no references here. I sort of missed.
Well, assuming people put this into Google, just the title.
Okay.
Yep.
That will do. Okay.
And finally, some acknowledgments. So this whole project has been led by Debbie Marks, who set out to solve this in 2010, and it's been quite a story since then. Chris Sander, who's also worked on this, thanks Burkhard for making all of this possible, and thanks to Charlotta, who worked on the protein complex paper with me.
Okay, questions?
I just have one small doubt. This second method, the one which was working in the end, we didn't use chemistry at all. It was still totally statistical.
It was just the 20 symbols. So
Yes,
the one that's
maybe the words chemistry.
yeah.
I mean,
So maybe you go.
there was like a lot of math which I obviously didn't understand. And
Let me
then
see.
there was something else.
That's what he calls chemistry.
So you meant on
And then you said there was
This one?
No, go further, just a couple of steps. No, not backward.
forward.
Yeah. Forward.
Do you remember the number?
No, but you just passed it. You just wrote a sentence over there in which you said, and there was some other calculation to this method. Is that calculation chemistry related or what? This.
Oh, some other technical details.
Yeah.
No, it's not. So
So this is purely statistical.
it's purely statistical. So what you do is, for once you have to just correct for or avoid overfitting, which is a bit of an issue. So there is some overfitting penalties, and I didn't talk about, there is a second step in calculating these numbers. You have to fiddle a bit with these, but I just left them away for simplicity. But it's nothing major, so the basic idea is what's on the slides. Yep.
Does this replace sequence alignments or other comparative modeling techniques?
No, it doesn't. So sequence alignments as such are just the basis of this, right? And with regard to comparative modeling, they're operating in very different parts of the business in a way. Because comparative modeling is great if you already know a structure that is related closely enough. So if you have a structure that's, let's say, 80% away in terms of sequence identity from your protein of interest, don't use this. Use comparative modeling, because it will give you a more reliable answer in a way. You can get additional information from this that you don't get from comparative modeling, so it may still be worth using this, but in terms of just the structure, use this. Use comparative modeling. But there are proteins where you just don't have a template, or the template is very far away. Let's say it's only 20% sequence identity. And now if it's only 20% sequence identity, chances are comparative modeling still gives you an accurate structure, but you can't be that sure about it anymore. But if you now, let's say, use a hybrid of these two techniques, so you do the comparative model, and you also use this, and you just integrate the information, or you compare, or you see, look where they differ. So you can just use a hybrid approach between the two and gain additional information. So they're not competitors or anything. They're more complementary because they look at the same or a similar problem from very, very different angles.
Let me just fill in sort of give say the same thing in a different context. Mm-hmm. So first of all, alignment. So that method typically it's 10,000 proteins aligned. Mm-hmm. Okay, huge alignment. So we need alignment methods. We need huge families. In fact, the 10,000 number in the first paper that you wrote for membrane proteins, there were 11 novel predictions. So for every 11 proteins the paper published, they gave a prediction of what the structure would look like that nobody had ever seen before. They would want to do 1100, but only for 11 did they have alignments that were big enough. So that is already, there is limitation of the method. You need large alignments, okay? With a growing sequence space, somehow you begin your talk, sequences hit us, you guys want them more, right? Yeah. So they are the most greedy ones who have ever shown up in this field of computational biology in terms of wanting more sequences. But alignment will always be the first step for them. The second issue really, comparative modeling, let's repeat that again. Comparative modeling is when you have a sequence of a protein of unknown structure, you have the database of proteins of all known structures, you map the unknown sequence by alignment to the known structures, and you in fact look up what is similar. If the sequence is similar enough, you say that my sequence of unknown structure has just that structure. You copy the coordinates. And that works for roughly every other protein in the database of the 55 million. For about half of them, we have one region where we can do comparative modeling. And I would just, what Thomas says at the end, try hybrids, is talk about what advanced researchers should do or may want to do. Or today, it's very clear. If you have that model, you take that model. But for all the other 25 million, or that is where this kind of method may apply. And again, it will not apply to all the other 25 million because you need huge lines.
Although, so the requirement in terms of the number of sequences is not as bad as it used to be, just because of this. There's been an algorithmic improvement 2 years ago, which allows you to have fewer sequences than originally.
Can you say what fewer is?
5,000 instead of 10,000. So there is it's hard to say what the actual number is because it really depends on what the protein family looks like. We've had cases, proteins of 200 residues, 300 residues. You have 1,000, 2,000 sequences and it works beautifully. You have other cases, you have 10,000 and it just doesn't work very well. So it's still necessary to have better metrics where you can use it and when.
Whoever listened to that, what he just said in the end. So the discussion is about how many proteins do you need in the alignment in order for this information. Carlo, you asked how can that work? And ultimately you have a lot of noise, and that's why you said how can that work? You have a lot of noise, and that means you need a lot of signal. So that means you need a lot of sequences. Now he says the number is not really the only important thing. Why could that be the case? So the number of sequences that you align. So again, if we went back a little bit in this talk here, this image really presents the idea. I believe this is the point that I mean. So what we're talking in this particular case here is the number would be 8.
Yeah,
So
but
you have
you're measuring
sequences.
frequencies in the end, so it doesn't matter, is it? That's the point.
Yes, but that is the issue of the signal to noise. So it's true that you only measure frequencies, but in the end, the more every single time for every pair you get some noise, and the more frequencies you measure, the more you get a signal over the noise. Yes, you increase the noise, but the signal increases more consistently than the noise. That's the reason why it works at all, and that's the reason why they need so big numbers. And again, so here the number is 8. We just discussed whether the number should be 5 or 50,000 or something like that. And Thomas said that sometimes 5,000 or 3,000 may be enough, in other cases 10,000 may not be. What could you imagine could be the reason for that?
The signal is weaker.
That's, yes, that's a very conservative, it's a great answer. Again, if you write an exam, that's a great answer, because it's a scientifically correct statement. The signal is weaker. Or it's not right. It requires that you understand the problem. And in that sense, it's a great answer. It's not only gaining time and space, but it's actually not explaining.
But you're asking why
more.
the signal is weaker, right now.
Why is the signal weaker?
Could it be that you have more of these transitive pairs?
That could be one reason. Exactly. But you think about other reasons. That would be immediately one explanation, yes. And that would actually be, again, in the context of an exam, that would be a high rated one, because you really have to understand something that he said, something specific that he said on the problem of that method.
We also said we need some kind of stability to have these couples to interact. Maybe one protein with loose stability would be much more difficult to. You need much more things to have correct data.
So now we talk about something that has to do essentially, in some sense, if you want, with the movability space. So your particular sequence has a particular structure, and that particular structure has a certain dynamic constraint. So it can move only to some extent. That's what you mean here with the word stability, and that's absolutely right. And it's not necessarily, not directly related to the structure, and it's not necessarily directly related to transitivity. Those two things may be somehow related, but they may actually be separate things. Yes, that would be another reason. Anything else more trivial? I'm talking about a number here. Remember the discussion we had about 85 million to 55 million? So just looking at the number, you don't consider how many of them are actually very, very similar to each other. So what you also need here is diversity. Right? And that is not that trivial to enforce. So they implemented a lot of, let's call it hacks, or more or less clever solutions. They and many people in the field have been doing that for 20, 30 years to somehow weigh the overall similarity. None of them really is completely solid and completely sound, and none of them really works. So that makes it difficult. So it's not a problem that is easy to solve. So they have the scale that they call the effective number. So rather, in some sense, the idea behind that, rather than counting the number of things you have here, 8 in this case, you would count the diversity in some sense. That helps a little bit better, but it's still not answering it because of these two reasons, and because of additional reasons that ultimately, I guess, pertain to the evolution of that family too. Okay, any other question to Thomas? If not, then we are free to have other questions, and you are free to go to your office.
Okay.
Okay, so let's get back There are a variety of things we can do for the remaining 25 minutes. One of those is that we say bye. What's that all? No.
Ja, oder ob das ja, je nachdem, was Sie haben, also zuerst mal die Frage, was Sie haben.
Pretty good.
Your mic is on.
Was? Oh.
Ja. Oder gibt es eine größere